All right, welcome everybody. So I'm re-recording. One of the slides was error on the previous recording. I rolled with it during the recording, but I figured I'd re-record it before most of you guys got to that slide and made it. This is already confusing enough as it is. It doesn't need to be anymore. So, all right. So we're going to start with population genetics today. And population genetics is simply a way of creating predictions from a set of data. The set of data that we're using is allele frequencies. And so if you think about what an allele is just, a variation of a gene, one of the variants of a gene. And if you look at how frequently that variant of the gene shows up in the gene pool, like in the population, you can use those numbers to predict what kind of genotypes you might see. Would I see a lot of homozygous genotypes or heterozygous genotypes? And then if I know how those genes behave when they go from the gene to the phenotype, then I can predict what kind of phenotypes are in the environment in this population, just from knowing my allele frequencies. You can also, and this is somewhat what this is used for more often, is you can work backwards. And so if you know what the phenotypes are in a population, which is a lot easier to do than know what the allele frequencies are, then you can sometimes work backwards and say, okay, well, if I can go out and count all of the different phenotypes, all of the homozygous dominant, all of the all the heterozygous, all the homozygous recessives, if I can count all of those phenotypes, provided that all three of those have a different phenotype, then I can work backwards and calculate allele frequencies from just seeing the phenotype. And that's where this is used more often. And so that's, but we're going to start the other way. So we're going to start with calculating allele frequencies and work it forward. And then at the end, we'll work backwards from phenotypes to frequencies. And so, why this is important? Well, let's get to our note sheet. So, because evolution is the change in allele frequencies over generational time, in order to understand how and why populations evolve, we must first be able to calculate allele frequencies. So, let's start with that. All right, so calculating allele frequencies. Allele frequencies are just simply the proportional number of a certain type of alleles in a population. And proportional just means what fraction of the total alleles are made up of a specific type of allele. So in a population with two alleles, let's say that those two alleles are big A and little a, the proportion of big A alleles is calculated by counting the total number of big A alleles and then dividing by the total number of alleles. So let's think about that. Like, let me use like a real world example for this. So if let's say that you have a bag of M&Ms and it's got 100 M&Ms in it. And let's say that 40 of them are red and 60 of them are blue. So they're the red and blue M&M bag, whatever that is, like Mint Crunch or something. So red and blue M&Ms. So there's 100 in the bag. 40 of them are red and 60 of them are blue. So what we would do is we'd say, okay, what's the total number of red M&Ms in the bag? And I go and count. I say, okay, there's 40. And then I say, well, what's the proportion of red M&Ms in that bag? And you could say, well, you take the number of red M&Ms, which is 40, and then you divide it by the total number of M&Ms in the bag, which was 100. And so ergo, you get zero point 0.4 for that particular problem. And we'll bring this up in a second, but if there's only two alleles, right? If there's only two colors of M&Ms, and I know how many red M&Ms are, and I know how many total M&Ms are, I don't even have to count the blue ones. I know that there's 60 blue ones, and 60 out of 100 is zero point 0.6. And so if you notice that the two frequencies or the two proportions have to add up to 1, and we'll get there in just a second. So in this particular case, we have incomplete dominance in this population. And so a big A allele is going to code for a big head. A little a allele is going to code for a small head. And if you happen to be heterozygous, you're going to have just a normal size head. Okay, so let's go count. Let's go count the number of alleles. So I'm out here. I do a genetic test on these people, and they all come back. And now I have my results of the genetic test, and I know that this individual has two big A's. He's homozygous dominant. This individual has one of each, so he's heterozygous. And then this individual is recessive, homozygous recessive. So let's just go and count how many alleles of each type we have. So of the big A, I've got one, two there, three, four, five, six. Let me use my pointer. Seven, eight, and then nine, 10. So I know I have 10 of the big A alleles. And then now I can go in and I can count how many little A alleles I have. I have one, two, three, four. Okay, I've got four. So how many total alleles do I have? 14. And so now from this data set, I can actually tell you what the allele frequencies are. So the first one, the frequency of the big A allele in this case is going to be 10 divided by 14. So, and then the frequency of little a would be 4 divided by 14. And so I get 0.71 is my proportion or my frequency of the big A allele. And then 0.29 is my frequency of the little a allele in this population. And so one thing you might notice is that add up the two proportions together, the two frequencies, and you will get 1. Okay. So in a population with two alleles, we can say the frequency of A plus the frequency of little a, big A, plus the frequency of little a must add up to 1. Okay, so let's talk about the Hardy-Weinberg equation. So Hardy-Weinberg is actually kind of a hyphenated thing. There's a guy named something Hardy and something Weinberg, and together they created a paper where they came up with kind of a general formula for how to calculate allele frequencies that instead of using like big A little a or big B little b, they use a more generic coefficient. They use P and Q, or I should say variable. They use P and Q as their two variables. And so you could say P is essentially equal to big A and Q is essentially equal to little a. And so according to Hardy-Weinberg, the frequencies for the two alleles, P plus Q, have to add up to 1. And so that's just what we saw earlier is the frequency of big A plus the frequency of little a have to add up to 1. Or, you could say it that way too. All right, so now we have to ask the question. So to figure out the frequency of genotypes, right, we're going to have to do something different here with the allele frequencies. So once we know our allele frequencies, once they're determined, we can use the allele frequencies to predict the frequency of each genotype in the population. And this is done by simply asking, what's the probability that I pull out a particular set of two alleles from the population? And it's 100% based on what the allele frequencies are. What you can do with Hardy-Weinberg, and you can do this with anything, you can do it with big A and little a too, is you square both sides. So essentially what you're saying is you're saying, okay, I'm reaching into this bag of M&Ms. I've got 40 red ones and 60 blue ones. What's the probability? I'm going to reach in there, and if I'm going to create an individual, I'm going to pull out two M&Ms at random. And so what's the probability I reach into this bag and I pull out two red M&Ms? What's the probability I pull out two blue M&Ms? What's the probability I reach in and pull out a red and a blue M&M? And so that's how we calculate the genotypic frequencies. Now, one thing that I'm going to notice here, right, is that you've got 2PQ in the middle. And so let's figure out where that comes from, because we understand P squared. P squared is the probability that I pull out a red one and then a red one. Q squared is I pull out a blue one and a blue one. But why is this 2PQ here? Why isn't it just PQ? Well, the answer to that is you can make a red and a blue combo two different ways. You can make a red and blue combo by picking a red one, then a blue one, or you can make a red-blue combo by picking a blue one, then a red one. So you can actually create a red-blue or a heterozygous two different ways. For instance, another way to think about it is dad could provide the big A and mom could provide the little a, or dad could provide the little a and mom could provide the big A. And those are two different ways you can make a heterozygous individual. All right, so that's another way to look at it. So one of the ways that we get this, and let me just make sure that I'm not skipping anything in our note sheet. Okay, so the genotypic frequencies for Hardy-Weinberg can be calculated by squaring both sides of the original equation. We use the FOIL method, so that's first, outer, inner, last. Hopefully that's still familiar to you guys. And then we get P squared plus 2PQ plus Q squared equals 1. So if there are two alleles, and we know, well, we'll get to that one next. Okay, so another way that we can look at this is we can say, all right, well, if I take P and Q and I line up kind of like a Punnett square here, then I can put them into this Punnett square. I'm trying not to get this, you guys probably can't see it, but I got this thing on my page that pops up if I move my cursor too low to the bottom. So anyways, we get basically I create a matrix, kind of like we do with our Punnett square. And in that matrix, I can see all the different combinations. And so I can see here that there are two different ways to make PQ. And so that's where I get the 2PQ from. And then I did it here with the big A, little a example. So just like Mendelian genetics, I put them into the matrix here, into our Punnett square-ish thing. And then I get, here's how I would calculate what the probability of getting a homozygous dominant individual would be. I would just take A, the frequency of A times the frequency of A, and that would give me A squared. And then that's my heterozygous, so 2 big A, little a, and then my little a squared would be right here. Okay, so here's our example. So if there are two alleles and we know the allele big A appears in the gene pool at a frequency of 0.45, then we know by definition that little a has to appear in the gene pool at a frequency of... So let's figure that out. How do we figure that out? Well, we know that the frequency of big A plus the frequency of little a is going to equal 1. And so I can write it into this formula like this, where I say, okay, here's the frequency of big A plus the frequency of little a has to add up to 1. And so I can just solve for little a. I can just subtract 0.45 from 1.0, and I get little a is equal to 0.55. And so now I know the frequency of big A is 0.45 and the frequency of little a is 0.55. And so I can ask the question, okay, well, what kind of genotypes and phenotypes can we expect to see in this population? So now that I know my allele frequencies, what kind of genotypes can we see? And so to understand how we calculate the frequency of each genotypes, we place the allele frequencies into a matrix, and that's what we're going to do here. And so big A represents the frequency of big A. Little a represents the frequency of little a. And I'm going to plug those two frequencies into my matrix here. So it kind of looks like a Punnett square, but we'll call it a matrix. This is literally how they do linear algebra, if you're curious about that. If you want to get into like what would that be, like Calc 4 or something? Anyway, I digress. Not my specialty. In this table, we can see that so here's my gene pool. So in my gene pool, if I was to go in here and add it up, I'd count out the proportion of big A's to little a's. And then I could say, okay, in this gene pool, the frequency of big A is 0.45 and the frequency of little a is 0.55. Okay. So what's the probability I pull out two big A's? Okay, well that's going to be 0.45 times 0.45. That's the probability. So you can think about kind of like a coin flip, right? Is that big A is like heads, little a is like tails. If the coin is weighted and I'm going to flip it and get big A 0.45 or 45% of the time, what's the probability that I flip it twice and I get heads both times? That's how probabilities work. All right, so calculating the probability of generating a heterozygous individual from our gene pool, we must add the two probabilities together. So that'd be the probability of pulling a big A out first, followed by a little a. Add that to the probability of pulling a little a out first, followed by a big A. That's why the formula is 2PQ. Okay? All right, so... And then the probability of pulling out little a is 0.3. And then if we were to add these all up, just to check ourselves to make sure, these should all add up to 1 as well. So my predicted genotype frequencies are going to be 0.2, 0.5, and 0.3. All right, here comes the fun part. So now imagine. Well, we'll get to that in a second. So let's jump into this practice here, okay? So this is what we're doing. We're going to put in we've got our genotypes out here. Okay, so we got our big heads or capital A, medium or normal heads or big A, little a, and then our small heads are little a. So let's do this. So let's count up our alleles. So we've genotyped. Oh, so here's an example, right? So if we were to look out here in our population, we'd say, okay, I know that a certain genotype will make each one of these phenotypes. So now I can go in and genotype all these people. And so that's what you would do. You would go genotype them. Now I've got their genotypes. Now I can count their alleles. So let's do that. So how many little a alleles do we have? 2, 4, 5, 7, 9, 10, 11. I've got 11 little a's, and I've got 1, 3, 4, 6, 7, 9 big A's for a total of 20 alleles. And so now I can calculate my allele frequencies. So say the frequency of big A is going to be 9 divided by 20, and the frequency of little a, 11 divided by the total, which is 20. I get 0.45 and 0.55. Great. Okay, moving on. I can put those allele frequencies in the matrix, cross multiply, and that will tell me the probability of getting a homozygous dominant, a heterozygous, and a homozygous recessive. And so I plug them into my table. And then boom. So that's how we figure out or predict genotypic frequencies from allele frequencies. All right, so let's carry. Oops, left a little head floating there. So let's practice this. So let's say, okay, so what's the so in my next generation of people, how many people with big heads should I see? How many people with normal heads, and how many people with small heads? So let's say how many people were produced, how many offspring were produced by these people here. Well, let's say that these people collectively had 10 offspring. And so we're going to multiply 10 by each one of these. And so I should end up with 2 people with big heads, 5 children with normal heads, and 3 children with small heads. Okay. And so I see 2 with big heads, 5 with normal, and there's my 3 with small. I forgot to add that animation there on this guy. Oh, he's still hanging out there. Hey, buddy. All right, so let's start over. So we come back here. What are the allele frequencies? Let's figure this out. Okay, so let's figure this out. Okay, so what are the allele frequencies? Well, we've got, maybe I'll let you guys do this one on your own. I think that was kind of the goal here. Boom, boom, boom. So now you guys can practice this. So go in there and practice, and you can pause this video and see if you can figure out what the allele frequencies are just from looking at these genotypes that were produced. Pause it and carry on. All right. So why didn't the allele frequencies change? What's up with that? I mean, if you look at the people, you go back and look at the people, right? I've got 3 big-headed people, 3 normal-headed people, and 4 small-headed people with these frequencies. And then I went and did this whole thing, produced some offspring. The allele frequencies didn't change, but the genotypic frequencies changed. That's interesting. So why did that happen? Why didn't the allele frequencies change? Well, the answer is that we're assuming that we're making some assumptions in this model. We're assuming that everybody mated randomly. We're assuming that all of them produce the same amount of children. And we're assuming a lot of other things too. And so nature doesn't necessarily work that way. And so what we'll get to, and we'll jump to this in a second, is we'll get to that there's some assumptions that when they're violated, we see allele frequencies change. And when we see allele frequencies change, that means populations are evolving. And so what we can do is we can trace our steps back and say, okay, what assumptions were violated that caused these allele frequencies to change? And that will give me a clue as to how and why this population is evolving. Okay, so what do you do if you can't count allele frequencies directly? And so that's the last paragraph on this first side of our note sheet. Now imagine that counting alleles is more challenging because one of the alleles is expressed with complete dominance. So what does that mean? Well, that means that there's going to be 2 genotypes that have the same phenotype. So that means that the heterozygous individual and the homozygous dominant are going to have the same exact phenotype. Look at them. They both have big heads. Well, how are we going to count those allele frequencies? If we go out in the population, we see a big-headed person, we're not going to know. Now, some of you guys that are pretty savvy are going to be like, wait a second. Couldn't I just take these individuals and back cross them to the recessives, doing a test cross like we did in Mendelian genetics? The answer is yes, but that takes a long time, especially if you're gestating a human, which takes 9 months. Plus, then you have to you've got the ethical ramifications of forcing people into copulating and all that stuff. So that's, in this case, it's not possible. But there's other ways to figure this out. And so what's nice is even though 2 genotypes share the same phenotype, we still have this individual here where their phenotype is obvious to us in the population. And if we know their phenotype, we know their genotype. And this is our Q squared. All right, so what can we do? We're looking out in this population. How many of these guys are heterozygous? How many of them are homozygous dominant? Well, the good news, where are we at? We're flipping the page now on our note sheet. The good news is that allele frequencies can be estimated from the remaining data. So homozygous recessives, these small head people, are easily identified because their phenotypes are different. All right, so by counting up the individuals that have the recessive phenotype, we can do something pretty cool. So here we go. So remember this formula here. We've got p squared plus 2pq. Well, the p squared plus 2pq, that represents all the big-headed people. And I can't isolate a variable and solve it there. But I can solve for the variable q if I know what q is. So I know that 6 out of 8 people are those, but I can't solve for those variables. But 2 out of 8, so 2 out of the total of 8 have q squared. So what I can do is I can solve for q. So what we can do is we can take the number of individuals that have the recessive phenotype and divide it by the total number of individuals, that would be 8. And that gives me the proportion of individuals that are q squared. And so q squared is equal to 2 out of 8, which is 1/4 or 0.25. And taking the square root of 0.25 is easy. You take the square root of 0.25 and you get 0.5. And so this gives me an estimation. It tells me that q could be 0.5, and that would, I don't know why I put a down there, it's dumb. And p would be 0.5. You know what I mean by that. So Q would be 0.5 and P would be 0.5 in this case. All right, so what are the estimated allele frequencies for big A and little a in this population? Well, big A is 0.5, little a is 0.5. So this little a over here should say P. Sorry about that. All right, just to make sure. So we counted the number of homozygous recessive phenotypes. We divided that number by the total number of individuals. That gave us 1 out of 4 or 2 out of 8 or 0.25. We know that the frequency of q squared genotypes is equal to that, and then we can take the square root of that number to get the estimate of the q, or the recessive allele. And then using the Hardy-Weinberg equation, we solve for the other allele frequency. Beautiful. All right, so here we get into this null hypothesis thing. All right. Hardy-Weinberg equilibrium is achieved when allele frequencies do not change over generational time. When does that ever happen? Never. But that's our null hypothesis. So the Hardy-Weinberg equilibrium is where populations do not evolve. And in order for populations not to evolve, these 5 things have to happen. There can be no natural selection. The environment cannot select better phenotypes for survival and reproduction. 0. Yeah, that would keep allele frequencies the same. Yeah, so natural selection would not change allele frequencies over generational time based on their adaptive value. There would be no migration, so that means individuals couldn't just up and leave the population, and individuals from other populations couldn't migrate into that population, because that would change allele frequencies. No mutation. So again, remember, these are part of the null hypothesis. So this is what we are trying to reject eventually. And once we figure out, if we see allele frequencies changing, we know it's going to be due to one of these violations of Hardy-Weinberg. So no mutation. Mutation is where one allele mutates into another, thus altering allele frequencies. Random mating. So random mating means that there can be no selection for mates that have higher reproductive value. That would increase certain alleles and allele frequencies if mates were choosy. So random mating, you might say, like, okay, when would random mating ever happen? And the answer is there's actually quite a lot of organisms on this planet that randomly mate. It's more often than not, that's the case. And usually when you have complete random mating, you have 0 parental care for the offspring. And so that's sort of like trees, flowers, those are random because bees just pick up pollen and spread it randomly. You got things like a lot of the sea life is random mating. Not necessarily like bigger sea life like fishes and stuff, but things like sea urchins when they reproduce, whew, they literally just blast a cloud of sperm into the water column, and then the other ones blast the column of eggs, and then the eggs and sperm meet out in the ocean and fertilize, and then you got your little zygote that develops, and it's crazy. Sea urchins are radial symmetric. They have radial symmetry, but the zygote that develops is bilateral, and then it becomes radially symmetric over time, which is like mind-boggling. Okay, I digress, but that's random mating. So random mating is like no selection for mates. And then finally, like Hardy-Weinberg also assumes, this null hypothesis assumes that there's an infinite gene pool, there's an infinite size population. Why is that important? So the larger the population, like let's say you have an infinite population, if somebody dies because they cross the street and get hit by a car, well, then it doesn't change the allele frequencies. You have an infinite population size. So random events cannot affect allele frequencies over generational time. Another way to look at this too is like if you have a really small population, if something happened, let's say you have like 10 people, okay, and like kind of like those big-headed people that we were just talking about. So if you have a population of 10 people and you count their allele frequencies and calculate them out, and then one of those people gets hit by a cattle car driving down the freeway, done, then now you only have 9 people. You lost 10% of your population. You lost 10% of your alleles. If that individual is homozygous dominant, well, now you lost like 10% of your homozygous dominant alleles. That's not good. So, anyways, smaller population, the more random chance can affect changes in those allele frequencies over generational time. And so, this is our null hypothesis. This never happens. Let me just be very clear about it. There is no world in which no natural selection, no migration, no mutation, complete random mating, and there's an infinite population. That doesn't happen. But what this means, since this is our null hypothesis, the null hypothesis is always that there's nothing going on. There's no. So here's an example of the null hypothesis in science. You could say like, okay, I just developed a drug that treats high blood pressure, and I'm gonna go distribute this drug and a placebo to a bunch of applicants in my clinical trial. The null hypothesis is that there should be no difference between the change in blood pressure between the group receiving the medication and the group receiving the placebo, that they would change exactly the same. That's the null hypothesis, that there's no effect. When you do see an effect and you see that the blood pressure medication lowers that one group's blood pressure by 20 points over 6 weeks, then you can statistically say, oh, okay, the chance that sorry, I bumped my mic. The chance that the difference I saw between the two groups, the chance that that is due to just randomness is very, very small. And so what if that chance is very, very small, then I can reject my null hypothesis in favor of my alternative hypothesis, which is that my blood pressure medication actually works really well. And so we're going to get into p-values this module when we talk about what causes variation in plants. Except we've kind of flipped it around on itself when we talk about p-values there. I won't get into that too much. Okay. So if Hardy-Weinberg isn't realistic, then why do we even bother using it? So when a population is in Hardy-Weinberg equilibrium, the allele frequencies stay the same. And so if we do see that change in allele frequency, it's assumed that one of those prerequisites on this page were violated. And then we can go investigate and say, okay, which one was violated? We say, is this population like evolving due to natural selection, or is there just a lot of migration happening? And so we can see which one was responsible for the change, and it gives researchers an idea of what's causing the evolution of certain populations. And then we can go investigate, and if we need to, we can take action. And so, yeah, that was the last line actually of our note sheet, because most populations non-Hardy-Weinberg equilibrium, we investigate to get an idea of how and why certain populations are evolving. Cool. I hope you guys enjoyed this. There's a worksheet that goes along with this particular lecture and subject matter. It does have a walkthrough video that goes with it. I highly recommend you guys. I'm not an idiot. I know that you guys can just like skip to the end and see all the answers and just copy them down. And if that's what you want to do, that's fine. Like I can't stop you. We're online. But if you really are trying to learn this and not just get accolades to put on a resume, if you're really trying to learn this stuff, like try doing the worksheet on your own and then watch the walkthrough and check your answers. And then that way you can see where your errors are in thinking and it will just help you learn things better. But that's just a recommendation. All right, guys. We'll see you for the next video. Bye.